later. So again, this is Analytical Chemistry or Chem102, another branch of chemistry. So you are done with Chem101. You have you had learned already some concepts about inorganic chemistry and also with organic chemistry. So hindi ko na ito i-discuss ha kasi I think nag-start na kayo nito. Like when you have two people in the bar, the other one will ask for H2O, the other one is H2O2. You know the difference? H2O is syempre water, H2O2 is hydrogen peroxide, which is used as an antiseptic. When you say antiseptic, that's for that's either to kill the microorganism or to prevent the growth of microorganism, especially in living tissues. That's the difference between antiseptic and disinfectant. Now for chemistry, we all know that chemistry is the study of matter. And you had your inorganic chemistry last time. When you say inorganic chemistry, you talked about the elements, their properties, their characteristics and behaviors, as well as the compounds of these elements. Now we have a special branch which is organic chemistry that deals specifically with carbon, since carbon behaves differently from all the other elements. So that includes also the study of the compounds containing carbon. So those two were already done last semester. Now again, in this semester, we will be talking about analytical chemistry, which I will discuss later. In your second year class, you will have your biochemistry, and bio means life, meaning you will be talking about all the chemicals that make up life, most especially those macromolecules which we can see in our body. So for example, carbohydrates, the proteins and the lipids, all of those will be discussed sa biochem. And then again, that will be in your second year. And then we have the physical chemistry that deals with the study of the physical properties of matter, but I don't think that meron kayong ganitong subject. Meron ito sa pharma, pero I think sa medtech wala na. Okay, now a little review about your lesson sa Chem 101. Syempre, we dealt most likely with periodic table. And as you can see, we have here a specific arrangement. The elements in the periodic table are arranged in increasing atomic weights as well as atomic number pa ganon. So itong mga elements na ito, remember, are grouped according to their properties or their behavior, and they are being color-coded depende kung anong group sila na belong. So group 1 here, naka-yellow. Ano nga ang tawag natin sa group 1? What do we call the group 1 elements?
Alkali metals po.
Okay, very good. The alkali metal. So everything is metal. Actually included si hydrogen dyan sa group 1A kasi meron din siyang 1 valence electron, but it's not really a metal. Group 2, how about group 2? This is called
alkaline earth
metals.
Okay, very good. Alkaline earth metals. So Bemagka-Cerbera pababa are alkaline earth metals. The elements here at the center are called transition metals, actually including those na nasa baba, but we specifically call these two rows here as rare earth metals. From the name itself, we can rarely see them sa earth. And then we have group 3, the boron group. Group 4, we have the carbon group. So this is the reason why we have organic chemistry. And then we have the nitrogen group sa group 5A. And then group 6A are the chalcogens, or these are the group of oxygen. I mean, this is the group of oxygen. And then itong naka-blue dito, not including hydrogen, we call them halogens. So do not forget, from fluorine pababa, we call them halogens. And lastly, Itong pink na ito, we have, pink ba yan? Group 8A, the noble gases. So they are called noble gases because they can stand with themselves, and then they are stable because they have completed the octet rule. So we don't really see these elements combine to form compounds. So we have talked about the parts of the periodic table. So don't forget ha, pag horizontal, pag horizontal, we call that period, and then the grouping na vertical, patayo, we call them group or family. Okay, and then if you look at your periodic table there, you can see a lot of numbers aside from the name of the elements and also the symbols. So the one that is most likely smaller is called the atomic number, and that represents the number of protons. So do not forget that the number of protons ang nire-represent ni atomic number. Now for an element that is neutral, meaning walang charge, the number of protons is equal to the number of electrons. So this 11 here can also represent the number of electrons. Again, that is with an exception if the atom becomes an ion. Pag nagkaroon siya ng charge, dyan magbabago ang number ni electrons. And the larger number here at the bottom is your atomic mass. And then again, as a review, this represents the number of protons plus the number of neutrons. So ito yung makikita natin sa periodic table. Aside from that, may iba pa. Depende sa periodic table na hawak nyo. But itong na-highlight dito, those are the important things that you have to be familiar. Sa analytical chemistry, from prelim up to finals, we will be dealing with problem solving, computations, and so on. So please be ready with your calculators. Kasi as in, magsosolve talaga tayo. And among the information you can see in the screen, the most important thing that you need to know, syempre the symbols, the name of the elements, and this atomic mass. If you have learned how to compute the molecular weight, dati sa Chem101, babalik pa rin yun siya. So thus we need the atomic mass or the atomic weight. Okay, now let's define what analytical chemistry is. This is a branch of chemistry that deals with the composition and structure of matter. So we will be investigating again the composition and at the same time the structure of matter. In the investigation that involves every process and at the same time the gathering of the data, but hindi doon magsa-stop. Scientists are obliged to communicate the results of all investigation. So whatever we found out during the investigation of the composition and structure of matter should be kailangan siyang i-disseminate. Okay? Now, since analytical chemistry is more on problem solving, then we have here the analytical approach on solving problems sa chemistry. So step 1, syempre, we have to identify and define our problem. This would maghimog investigation if we don't really know what exactly is our problem. So we have to deal with the problem's context, and at the same time, in the beginning of the investigation, the type of information that we need should be clarified. Kailangan na-clarify yon at the beginning of the investigation. And then after that, step 2 will be the designing of the experimental procedure. So we don't really have to make our own experimental procedure. We can adopt yung mga standardized procedure that we have in chemistry. But syempre, we have to make sure first that the procedure that we are going to use is valid and at the same time suitable for the problem that we have at hand. So we also need to establish criteria. We also have to identify potential interference or interference. Interference are those chemicals or things that can hinder us from getting a correct result. So for example, for a chemical compound, if a chemical compound contains contaminants or impurities, those things become interference. So we have to identify first whether we have the presence of those things before tayo mag-move on with the experimentation. So again, we have to establish validation criteria. We have to select the most appropriate method and establish sampling strategy. So we do sampling here. It doesn't mean na naakay isa ka drum nga chemical, all of it will be used during the investigation. Syempre, we will just get a little amount of that, and that's what we call sample. And at the beginning, we have to establish how are we going to do the sampling? How are we going to select the small portion that we are going to use in our study. Now, if everything is prepared, that's the time we conduct experiment and gather our data. So that is through laboratory experimentation using calibrated equipment and instruments, and at the same time, standardized reagents. So when we say standardized reagents, we know the concentration, we know the real concentration, and we have established the purity of the chemicals to be used in the experiment. So when we do experiment, syempre we gather our data, but that it does not end there. Syempre we need to analyze our data, and we also have to interpret it. After interpretation, that's the time we know whether the result of the experiment is sufficient to answer our problem. Or if ever hindi, syempre may new problem that will arise, and that we will go back to step 1. So these are the analytical approach to solving our problems in analytical chemistry. Now, I don't know if na-lesson na ito ni Ma'am MJ sa inyo, but if ever lang you will become an analytical chemist since you have this subject in college, pwede nyo din naman maging work ito aside from a medical technologist talaga, na kung ano yung job description ng medtech, ganun talaga ang gagawin ninyo. Aside from that, you can be an analytical chemist. So if you opt to become one, now this will be your job function. So sige, number 1, perform qualitative and quantitative analysis. So we will differentiate the two later. Kung ano yung difference ni qualitative and ni quantitative analysis. Now, I told you a while ago that we don't really do testing for all the chemicals in the laboratory. Instead, we just get a small portion of it, and yun yung igagamitin natin sa investigation. Therefore, the analytical chemist should know how to do the sampling, how to define the sampling, isolate it, concentrate, or even preserve our sample. Now, the analytical chemist will also set error limits. Analytical chem is very strict class when it comes to measurement. Thus, the analytical chemist will also define the boundaries when it comes to the standards and the specification. So if it goes beyond the standards and specification, error na yun na part. Okay? Okay, sige, Mr. Amadeo. And then validate and verify results through calibration and standardization. So nasabi ko kanina, analytical chemistry is very strict sa measurement. Therefore, it is also the job of the analytical chemist to make sure that all the instruments and the equipment in the laboratory are properly calibrated. So basig ano na siya, nagtaya-taya na or wala na calibrate, for example, the analytical balance, hindi na siya magbibigay ng tamang mass. So dapat pag-start pa lang ng experiment, naka-calibrate na yon, and that all the chemicals to be used, all the reagents to be used in the experimentation are also standardized. So tama yung concentration na naka-label. That's what we call standardization. And then perform separations based on different chemical properties. We have a lot of mixtures in the laboratory. Remember, mixtures, pinaghalo-halo na yung mga compounds. And then, if ever ang kailangan lang natin is i-assay or mag-investigate tayo ng isang component lang ng mixture, dapat yung analytical chemist alam niya paano i-separate yung mga components ng mixture na yon. Okay, and then create new ways to make measurements if ever the existing methods are not really suitable. And then interpret data in proper context. And lastly, communicate the results and the conclusions of the investigation to other scientists. So if you want to be an analytical chemist class, please take note of the following functions that you may perform if maging analytical chemist na kayo inside the laboratory. Okay? So now, questions so far before tayo mag-move on with the divisions of analytical chemistry. Meron bang gustong i-clarify? Wala? Sige. Let us move on with the two divisions of analytical chemistry. Analytical chemistry is also known as qualitative, quantitative chemistry. The first division is called qualitative. Qualitative because this is a branch of analytical chemistry that deals with the identification of the composition of matter. So whether that is a mixture or a compound, we will be identifying what is in that chemical substance. And we will also be identifying whether there are impurities or contaminants present in our chemical substance or our reagent. So this branch will answer the question, what is present in a given sample. Like I am going to give you a chemical or a metal na solution. You will identify anong klaseng metal ang nandoon sa solution na yon. So if you can still remember the flame test na hindi naman talaga na-perform kasi di tayo nag-face to face, pero na lesson naman, di ba yung flame test class, you have a chemical substance, you subject it sa flame of the Bunsen burner, to be specific, the non-luminous flame sa Bunsen burner. Remember, di ba, it will give you different colors sa flame. So different metals, different colors sila. So that is what you call a qualitative testing. You will just know what is present in that solution. Diba? If it gives you a yellow color, then that is a sodium solution. If it gives you a purple flame, then that is a solution of potassium. So na-answer mo what is present in a given sample. That is what you call qualitative analysis in analytical chemistry. But if we really want to know how much is present in our sample, that's what we call quantitative analysis. Meaning we will really identify here the amount of the substance, the amount of a certain component sa ating chemical mixture. Okay, again, this division answers the question, how much is present in the sample. So for example, sa flame test kasi hindi natin alam gaano karaming sodium ang nasa solution na yon. Ang alam natin, meron siyang sodium kasi nag-yellow yung color ng flame. Pero pag talagang pinagawa sa iyo quantitative analysis, then you will have to subject that certain solution to a specific testing for you to know how much sodium is present. So you can use machine or equipment in the laboratory. I'm not sure if you are familiar, but if flame test will let you know that sodium is present in the sample, then if you want to know the quantity, then you may use this equipment in the lab. Atomic absorption spectrophotometry. So this one class is a machine na if you will place the solution inside, you will really know exactly how much sodium, for example, is present in your sample. But actually, yung concept niya, flame test pa rin. Naging sophisticated lang siya na instrument kasi pati ang quantity malalaman mo. So ito yung ginagamit class nila to test kung gusto sila kung gusto talaga natin na malaman kung gaano karaming metals ang present sa sample, ito yung ginagamit nila na machine. For example, girls, yung lipstick natin, 'di ba? Yung lipstick sometimes it has lead, the metal lead na content kasi yung lead pampatingkad yan ng kulay. So for example, sa lipstick. However, too much lead in the body will become toxic. It will be poisonous. So kung gusto natin malaman kung gaano karaming lead yung isang lipstick, isa-subject natin siya sa atomic absorption spectrophotometry. Okay, I hope nasabtan to siya. Okay, sige na din. Yan ha. So, hinay-hinay girls sa paggamit ng lipstick. Kasi, ano kasi yung lead is called cumulative poison. So, it will not really show toxic effect agad-agad in the body. The lead should accumulate first in the body before it will really give us that toxic effect. So, dili basta-basta mo palit o lipstick. How do you know nga na ay lead? Kaya syempre sa atong balay, wala man tay atomic absorption spectrophotometer. Wala man tay ingana sa atong balay. Paano kaya nato mabalaan? Dili siya quantitative, qualitative analysis lang, nadaghan og lead ang atong lipstick. How do you, alam nyo paano? You have an idea? Wala? Kuhanin nyo inyong lipstick. Tapos, butang ninyo sa inyong kamot ang inyong lipstick. And then you get kanang alahas ninyo nga gold. Pero ang gold jud ha, dapat gold jud siya. Dili siya fancy, dili po siya silver. Gold dapat jud. Tapos i-brush ninyo inyong gold na any nga ano ba na siya, kanang ring ba na siya or jewelries ninyo, basta gold, inudnod ninyo dito sa inyong gibutangan og lipstick. Pag mo itom gani na, ang example, ang lipstick ninyo red, pag human ninyo og brush sa inyong jewelry, kay mo itom na siya. Meaning ana, daghan og lead ang inyong lipstick. So you try that para kabalumog. Kabalumog, daghan jud bag lipstick or gamay lang ang inyong daghan og lead inyong lipstick. I-try lang. If ever meron kayo dyan jewelry na made up of gold. Basta na ay gold ha. Magamay man na or madako iyahang karat. Basta na siya gold. Mo-react na siya sa lead. And then, mo-itom na inyong lipstick. Sa kamot ibutang. Okay? Questions so far? Wala? Sige. So let's move on with the term analytes. Let's just have definition of terms. When you say analytes, it's the component of the sample that you want to determine. Pareha sa example nato kagaina, lipstick. Remember, ang lipstick marami yan siyang component. isa lang yung component na lead dyan. The rest, marami pa para mabuo yung lipstick natin. But in our case, sa investigation, ang gusto lang talaga natin malaman, lead lang. So lead yung component ng ating sample that we want to determine. And we call that an analyte. The lead there is called analyte. Kasi yun yung ang focus ng investigation natin. We don't want in that case we don't want to know the other components. We just want to know how much lead is present in the sample. Analyte ang tawag doon. Yan yung focus of the investigation. Now, sa chemistry and even in your research, let me just get my pen so that I can write sa screen. Mga investigation class, whether it is in the chemistry laboratory or when you do your research or your thesis, either we do replication or we do trials. Ang importante dyan, hindi lang kaisa tinitest yung sample natin. Ano ang difference ngayon ni replicate and ni trial? Pag replicate class, we have same sample. So example class, lipstick, no? So kukunin mo siya sa isang lipstick lang, a little portion. And we do the testing using the same method. And at the same time, we do the testing at the same time. So for example, the first portion taken, for example, from the lipstick is done at 9 a.m. The next portion should also be done at 9 a.m. So dapat sabay talaga silang ginawa. The same method, the same sample. So meron kang replicate 1, replicate 2, or replicate 3. And you will have results for this. So for example, ang nakuha mo sa replicate 1, meron siyang 10 grams ng lead. Ito merong 9 grams ng lead. This one contains 9.5 grams ng lead. We will just get the average after. So we are doing replicate samples para makita natin ang precision ng ating results of the experiment. So again, we don't do one testing only for our sample. Either we do the replication or we do the trial. So how about the trials? How do we do this? For trials class, we still have the same sample. So this will be taken in one product or in one chemical substance. Pag fake ang gold, dili na siya mag-react ayan. Dapat dapat tinuod jud na siya na gold para mag-react siya. Pag daghan kaayo na siya og lead mo itom na inyong lipstick nga either pink or red, mahimo na siyang black pag daghan siya og lead. That's how we test kasi wa man tay AAS atong mga balay. So again, sa trial, same sample, same method, but not the same time. So example, you carry out the experimentation for the first portion na kinuha mo doon sa sample mo today, tapos na kulangan ka ng time, so bukas na lang yung iba, trial na ang tawag doon. We cannot call it replicate anymore. Replicate is done at the same time. Okay? Please take note of the difference between the two. Kasi yung iba, syempre pag nagkakandak na ng thesis, minsan na i-interchange yung term na replicate or yung trial. So pag inask kayo ng inyong panel sa research, how many replicates na ginawa ninyo, kailangan dapat sabay-sabay talaga yun pinerform. Pero pag hindi nito sinabay, sabihan nyo lang na trial ang ginawa nyo, hindi replicate. Okay? Hope na yan siya, na-clarify yan siya na meaning ng dalawang terms. Now, let's move on with another term that we may encounter in analytical chemistry. We have here interferences. I have actually mentioned this a while ago. When you say interferences, that's any species, any chemical component that may give us error during the analysis. So as much as possible before conducting the experiment, we have to prevent this error from happening. We can either get rid of the interferences or we can minimize their amount, or we can actually mask them. So there are procedures na ginagawa para ma-mask ito. Kasi remember, sabi ko, di ba, sample sa lipstick. Kung gusto natin malaman yung amount ng lead dyan, remember, ang lipstick, it's a mixture. Maraming component ito before nabuo yung lipstick na yan. But in the analysis of lead, the other components of this mixture may actually hinder us from getting the correct result. So merong mga procedure sa analytical chemistry na ginagawa either to isolate the lead alone or to just mask the other components. Pag sinabing mask, tatabunan natin sila na hindi sila ma-detect during the analysis, hindi sila magbibigay ng false result. Okay? That's what you mean by interferences. Questions so far with the terms that we defined before we move on with the grades of our chemicals. So even our chemicals have grades. Meron yan silang grades. May mataas na grade, meron ding mababa na grade for our chemical substance. Questions? Wala? Clarifications? Sige, let's move on. The grades of our chemicals. Yung mga chemicals natin, class, are being graded according to their purity, according to their quality. So the first one there is the reagent ACS. ACS means American Chemical Society. Again, American Chemical Society. If the chemical is being graded as reagent ACS, meaning it is high quality na chemical, it has high purity, and that it is suitable for laboratory use. So it can be called a high grade chemical if it is labeled as reagent ACS. So any chemical ito, whether it is a sodium chloride, pag si sodium chloride may nakalagay sa baba, reagent ACS, meaning that certain chemical is of high quality, that certain sodium chloride is high quality. Okay? Questions? Wait lang ha, replyan ko lang si madam. Sige. Next, we also have the so-called guaranteed reagent or GR. Again, guaranteed reagent or GR. Makikita nyo ito sa label. Basig matingala mo, paano na ito mabalaan, ma'am? Reagent ACS na siya. Makita na siya sa label. Example, this is your chemical substance. Nakalagay siya sa isang reagent bottle. Makikita yan siya sa label, class. Example, nakalagay dyan, hydrochloric acid. Ito yung chemical natin. Sa baba, may nakalagay dyan either reagent ACS or pwede ding nakalagay GR. Pag nakita nyo yung GR, that means guaranteed reagent. So pag GR, ang grade ng ating chemical, it's still suitable for analytical chemistry na use. And ano yung standards na sinusunod natin for guaranteed reagent? Pwedeng sa ACS na requirement, American Chemical Society na requirement, pero pwede ding mag-exceed pa siya with the requirements ni American Chemical Society. So if it is labeled as guaranteed reagent, it is also considered a high quality chemical. So hindi lang yan siya para kay hydrochloric acid, ha? Para din sa ibang chemicals. Okay? Now we also have AR grade. AR naman means analytical reagent. So this is more or less similar also to American Chemical Society na grade. Okay? Now, in the school's laboratory sa doctors, since wala pa man ta naka-visit yun sa mga laboratories, pero once na you will be allowed to do the face-to-face. Actually, we are allowed pero wala pa naman sigurong nag-start pa talaga na mag-conduct ng experiments sa school. But you will see class na yung chemicals na nakastock sa ating laboratory are actually more on AR grade. Naka-AR grade yun sila. So we can see the original label. May nakalagay talaga after the name of the chemical, merong AR. So again, analytical grade yun siya. This one is more or less similar to ACS. So that is also considered as high quality reagent. So dili ta magtaka class nga nung medyo pricey yung laboratory fee ninyo. I don't know actually the exact amount for the laboratory fee, but medyo pricey siya kasi we use high quality grade chemicals sa laboratory. Pag dili man gud siya high quality, remember there might be impurities and these impurities can become interferences during the experimentation. So wala siya nga gina-make sure. Ang in charge nato sa laboratory si Ma'am Charm. So I believe you know her kay siya man inyong teacher sa Chem101. So siya ang nag-secure og mga chemicals sa school and gina-make sure yun na siya na high quality para dili magkaroon og problem sa results sa experiment. Even if experiment lang na siya sa estudyante, dili siya experiment sa mga scientists yun sa mga big laboratories, pero dapat i-make sure man good nato na tama inyong makuha na result and you will really observe na tama ang reaction. Okay? Now for titration, the lesson ni siya sa Chem 101, ang titration. If we want to know the concentration of our substance, we use a primary standard. So this is also a high quality chemical but is specifically used during titration. Titration processes, any titration process. And then purified ang tawag sa chemical if it's still a good quality chemical but there is really no official standards. Wala talagang official standards for it na ma-make sure natin na 100% pure talaga siya. But good pa rin yung quality, wala nga lang standard. And usually ito yung mga inorganic chemicals natin. So again, chemicals diba without carbon are called inorganic chemicals. Most of them, ang kanilang grade is purified. But then again, good quality pa rin naman. And then we have lab grade. These are the solvents that we use in histology. What do you mean by histology? Histology, logy is the study. Histo means?
Tissues. Tissue, ma'am.
Very good. Histology is the study of tissues. Okay, Shan. Sige. So those na sa inyong I think humana mo sa inyong anatomy and physiology, no? Kapag nag-ano sana tayo doon, nag-laboratory tayo na face-to-face, merong mga solvents na gagamitin to process the tissues, the cells in the laboratory. Ang dapat ng chemicals doon na actually gagamitin is lab grade. And then we also have the so-called USP grade. USP, by the way, i-type ko na lang para madali atong live. United States Pharmacopeia. That's what you mean by with USP. United States Pharmacopeia. We actually have USP in the school. It's a book. It contains a list of the chemicals sa kalibutan and nakasulat din mga standards and specifications for those chemicals para sa ilahang quality. Now, if that certain chemical has passed the requirements of the United States Pharmacopoeia, then it can be called a USP grade chemical. Okay? So it is manufactured under CGMP. CGMP means current, small letter jud ng C ha. Hindi na siya typo. Nga na siya pagsulat. Small letter C. Current good manufacturing practices. That's what you mean by CGMP. So again, these chemicals are being manufactured following the current good manufacturing practices. Okay. Any questions so far? Wala na. Sige. Continue ta kay murag lamig gyud kayo itulog sa ulan ba na adjoy sige disconnect ani ron. Kay hinay jud ning internet. But anyway, this one is being recorded. So you can review the discussion anytime. NF grade. NF grade ang chemical if it also passed the requirement of the national formulary. We also have a book of this in the school. Again, we call that national formulary. The same with the USP class. It also contains chemicals and the standards and specifications for those chemicals. Therefore, if it is being manufactured and past the requirement of the national formulary, it can be called an NF grade chemical. So both USP and NF grade chemicals are also considered high quality. So, puros high quality atong istoryahan, no? Gusto mo tanong, paunsa ang low quality, ma'am? Asa karon ang low quality? The low quality can be called the technical grade. So, the technical grade chemicals are suitable for general industrial use. So, it might not be suitable for the analytical chemistry laboratory. So, let me give you an example. Ang mga estudyante last time nag-thesis sila. And then, syempre naa sila plant material. Now, ilahang plant material, kailangan na nila i-extract. And for the extraction, they need ethyl alcohol. Or we call this ethanol, di ba? Ethanol ang tawag sa kanya. Now, ana sila ma'am, magpalit daw sila chemical niya, nalibog sila sa ilahang paliton. Ana sila, naaday 1 liter nga sila gusto tag 150. Naaday 1 liter nga alcohol na tag 150. Palito na nila sa mga tagabaligyag chemicals sa. Tapos napod sila yung nakita na 1 liter na tag 1,600. Ano sila ma'am unsa mo paliton ani? Parehas man daw na ethyl alcohol or ethanol. So I told them this one is a technical grade alcohol. And I told them to look at the label for the 1,6. This one is the AR grade. So as you can see, since this is high quality, mas mahal po good na siya. And since this one is just for general industrial use, then mas barato po na siya. So sa ilahang thesis, I told them, you have the freedom whether to buy this or to buy this one. Kasi the purpose lang naman is for the extraction. Hindi naman siya for analytical chemistry. Okay, that's how you differentiate the different grades of our chemicals. Okay, questions? That's the end for the grades of the chemicals. Wala. Sure na yun. Kasi nabutan na ha. Yes, Mr. Amadeo, you're raising your hand.
For example po, ma'am, sa technical grade po, ma'am, tsaka sa AR grade, ma'am, same na po ang kanilang results po, ma'am.
Okay.
If they are used.
The same result. Actually, dako ang chance. There will be a higher chance for you to have a different result if you use technical grade or you use in the experimentation an AR grade. Kasi si technical grade, we are not sure whether it is pure, so it may contain contaminants. And these contaminants may become interferences, meaning they will hinder us from getting correct results. They may react with the other chemicals that we use during the experiment. Thus again, iba na yung kanyang result. That is why in the analytical chemistry laboratory investigations, we don't really use technical grades ng mga chemicals. Instead, we either use the GR or the AR grade. Para sure talaga tayo with the result. We actually had an experiment way back 2018, 2017 with my students. Titration yung ginawa namin. So again, titration ang ginawa namin. Tapos ang ginamit nila na chemical is technical grade. Naubos na lang nilang isa ka litro. Wala paghihapon nag-react ang ilahang gihimo nga experimentation. Nagikapoy na sila kay, di ba, syempre sa titration, wala man gudta sa school, magnetic stirrer. So kayo mismo yung magsi-steer using your hands. Gikapoy na sila, sige tuyok-tuyok sa ilahang Erlenmeyer flask, isa ka litro na ilahang nahurot. Ay, tapos nagdugang pa jud pa sila. Wala gihapon siya nag-react. So mauna siya nga, anak ko, dili man good pure ang inyong chemical and dili siya ka high quality. So mag-expect yun, magdugay yun na siya mag-react. Tapos murag na isa ka group nga na ang ilahang leader, gikapoy na yun sakit na ihang kamot. Na siya ma'am, giyabo niya tong remaining na, wala na niya gi-measure, remaining substance. Kaya dito pa nag-react ang ilahang chemical. Ako, wala na mo yung result, ana, kay wala naman nakabalo sa volume, kay mo naman ang giyabo diretso, na walay sukat-sukat. So, wala na sila nakakuha og tama na result. So, mo lang na ang problema kapag dili high quality ang ato ang gamiton. Okay? Anything else? Alay? Sure na good? So, let us move on. What time is it? I still have 30 minutes. By the way, you can eat while I am discussing here. You can eat your breakfast. Pwede ra mo mag-turn off sa inyong hangkam kadali and just be back after eating. Kay basig magutman taan ni diretso ato ang klase. So you can eat. It's okay, Miss Alabado. Okay, units of measurement and significant figures. Actually, you are done with this with your Chem 101. So, i-review ko lang siya. So, we have here, take note that the worldwide na accepted na units for our measurement is the SI, the International Standard Unit. So, take note that for the mass, it's kilogram. This is the definition. I will not really focus on the definition kasi this is how they just get na unsa na siyang kilogram, unsa pud ng meter. As long as we know that it is the unit of measurement, then I'm good with that. So for the distance, it's meter ha. Ito lang definition niya. Paano nila nalaman na meter yung sa distance? It's the distance light travels in this 299,792,458 to the -1 na exponent na seconds. So yun yung definition ni meter. Pero murag buwang na tas gig-memorize ana. Dili lang ta mag-concentrate class, measurement and then the unit and the symbol. And then for temperature, remember it's Kelvin. It's the SI unit because it has the absolute zero. Si Celsius kasi and si Fahrenheit don't have absolute zero. Di ba natay negative 40 degrees Celsius. So walay absolute zero. For time, it's second. For current, it's ampere. And for analytical chemistry, most of the time, we will deal with the amount of substance. So you will always encounter the mole. The moles or the mole. Okay? And then other units that we may use also in our analytical chemistry na concept. For the length, no, this is an SI, not an SI unit, so we will seldom see this. We have the angstrom, and then the volume is liter, though it is not really an SI unit, pero parating natin yan siyang gagamitin. For force, we have the Newton, but minsan lang din ang force kasi ang force mostly kino-compute nyo sa physics. Pero I think wala na kayong physics except for those na nag-bridging last time. Pero sa mga succeeding na years, wala na ata kayo physics. Pressure also, sa physics din ito mostly. Pero meron tayo nito konti sa analytical chemistry. So we can use the Pascal. That's the SI unit for pressure or the atmosphere, ATM. Pwede din nating magamit yan siya. Now, energy, work, and heat can be measured using the SI unit, which is joule. Other than that, we can use the calorie and the electron volt. Pero actually, calorie is widely used even if it is not an SI unit. Remember, ang ato ang mga food products, when you look at the label, they use calorie or the kcal. Power is watt. Diba? Ito ang mga suga, naka-watts na sila. That's an SI unit. Charge is coulomb, potential is volt, frequency is hertz, and temperature, we also use most of the time the Celsius, though it is not an SI unit. Take note of their symbols, and you may also be familiar with the equivalent units for the conversion of our units. Now, aside from that, especially for the metric system, we use prefixes for exponential notation. Let me clarify this one. Dinudugtong natin ito siya class sa unit ha, mostly sa metric system. So for example, we want to measure mass. And for mass, we have the base which is grams. So dito yan siya, si grams nakalagay. Now, di ba meron tayong tinatawag na kilogram, milligram, and so on. Actually, ito sila lahat, pwede natin ito idugtong kay gram. Pwedeng yotagram, zetagram, etagram, petagram, microgram, nanogram, picogram, and so on. Pwedeng idugtong. And meron yan silang factor for the exponential notation. So for example, 1 yotagram is equivalent to, ito yung gagamitin natin, 10 raised to the power of 24 grams. So ito, iota, zeta, eta, peta, tera, giga, mega, these are used for very large measurements. Okay, very large. Because as you can see, 1 yotagram is 10 raised to the power of 24 grams. Pila na siya ka-million, billion na siguro. Okay? And then itong nasa baba, yocto, zepto, atto, femto, pico, nano, micro are also used for measurement pero sa mga smaller ones naman. Even the micro, di ba, hindi na natin siya makita with our naked eye. So very small samples na ito. Pababa ha hanggang yocto. Yocto is 1 yoctogram is equivalent to 10 raised to the power of 24 grams. Pero hindi lang yan siya sa grams dinudugtong ha, pwede din namang sa liter. Liter ang nandito sa gitna. So pag kiloliter, that means 1 kiloliter is equivalent to 10 raised to the power of 3 liter. Pwede ding microliter. So ang isang microliter is equivalent to 10 raised to the power of negative 6 liter. Ganyan yung meaning niya. Pwede din siyang idugtong sa seconds. Pwede din naman sa bytes. Yung bytes, di ba, sa storage information, yun siya. Storage of information. So dito sa gitna, yung bytes, meron tayong terabytes. Yan nata yung pinakamalaki so far. So 1 terabyte is equal to this one. 10 raised to the power of 12 bytes. Yan yung meaning niya. Pwede din gigabytes or megabytes. So, mga ano lang ito, prefixes, meaning dinudugtong natin sila sa unahan ng ating unit of measurement and meron silang corresponding exponential notation na equivalent. Okay, please be familiar with this. Daghan na kayo tag-isulod sa itong brain. Please be familiar. Okay? Any questions so far? Introduction pa lang ito, so I will not really give too much focus on this. Gagamitin natin ito as we go on with our lesson, especially sa ating mga computation. Karon kaya wala pa man tayo computation, so introduction sa heads up lang daan niya, kailangan familiar mo ani para dili ta magkalisod sa computation. Okay? Ala, sure na yun. We'll go back with the significant figures. Walang kamatayang significant figures. Wala question? Sige, let us just have 5 minutes na break, CR break. Be back 5 minutes after. Review lang tayo with the significant figures kay Chem101 sa LEC and sa lab, meron kayo nito. Pero in case lang nakalimot na. Okay, so brief lang ito. Number 1, non-zero. These are the rules. Non-zero digits are always significant. So lahat from 1 to 9, significant yan lahat ha, counted sila lahat. So if, for example, meron kang number na 24. Since both of them are non-zero, so definitely we have 2 significant figures here. Next, any zero between 2 significant digits are significant. So ito yung tinatawag natin na in between zeros kasi nasa gitna sila ng mga non-zero digits. So we have here 1,002. We all know 1 and 2 are significant and so does the 2 zero na nasa gitna. So all in all, we have 4 significant figures. So again, non-zero digits are significant. Next, lahat ng zero na nasa gitna ng non-zero digits natin are also significant. Pero yung zeros na tinatawag natin na leading zeros kasi nasa unahan sila ng non-zero digits, they are not significant. So just like this one, 0, 0, 5. We only have one significant figure here and that is 5. Itong leading zeros, itong nauna na ito are not significant. Even in the presence of the decimal point, as long as leading zeros yan, hindi pa rin sila counted as significant. So just like this, 0.5, kahit pa may decimal dyan, pero leading zero siya, 1 lang pa rin yung significant figure natin here. And that is for the 5. Okay, so any zeros after non-zero digits in a number without a decimal point are not significant figures. So meaning ito yung mga trailing zeros natin, meaning zeros that are found after the non-zero digit. So for example, meron tayo dito 20,000. One significant figure lang yan siya, class. Bakit? 2 lang ang significant. Kasi yung mga trailing zeros na walang presence ng decimal point, hindi sila counted as significant. Nagiging counted lang yung trailing zeros if merong presence ni decimal. So ito yung difference nila. Meron tayo dito 500.0. Pareha silang trailing zero pero may decimal. So counted ito lahat. Meron tayong a total of 4 significant figures here. So I hope na alala nyo pa itong rules ni significant figures natin. Okay? So let's do some exercise. Let's record this as your class participation. This will be the end of the chapter 1. So in the comment, kindly write the chemical symbols of the following. Comment section, i-1 to 5 nyo na para mabilis. 1 to 5 na, iisa nyo yung answer ninyo from 1 to 5 and I will record that for your class participation. So, number 1, cesium. Number 2 is gold. Number 3 is rubidium. 4 is arsenic. 5 is hydrogen. Let us see if you can still remember these are common elements. Let's wait for the others. Okay? Wala na? Or may hahabol pa? Let me see. Okay. Most of you got the correct answers for number 2 to number 5. Pero may problema ata tayo sa number 1. Check niyo daw sa number 1 kung sa tamang answer. Hindi nakapos. Tagay na. Lagi ulit ninyo inyong answer. Okay. Sige, let us see. Session is, syempre, CS. Wala, mali ang uban. CE inyong answer. This is CS. And then AU is gold. Rubidium is RB. Arsenic. Arsenic is AS. And hydrogen is H. So yung iba, medyo nagmali lang dito sa session. Kaya ang sinagot ay CE. So please don't forget that. Now, how about this? Write the atomic number of the following element. Ganun pa rin, isahin niyo pa rin yung answer ninyo. Hydrogen, ano ba atomic number ni hydrogen? Sodium, oxygen, carbon, and potassium. These are also common elements. Atomic number, ha? Dili atomic mass. Sige. So, let's see. Hydrogen class is 1. Sodium is what? That is not supposedly 23. Atomic number ang hinihingi, hindi atomic mass. Sodium is 11. Oxygen is what? 8. Carbon is 6. And potassium is what?
19.
This is 19. So, yung iba, ang binigay sa akin ay atomic mass. Remember, they are two different things. Si atomic number, kung nalilito pa rin kayo kay atomic number and kay atomic mass, the best thing is mas maliit si atomic number kaysa ka atomic mass kasi si atomic number ang nire-represent niya lang number of protons. Tapos si atomic mass mas malaki siya kasi aside from the number of protons, you still have the number of neutrons. So pag nalibog mo ha, kung pangitaan mo atomic number and atomic mass kay murag ang uban sa inyo ha, ang gisend dinhi kay atomic mass ni siya. Ang uban lang. Or the rest, you are correct. Correct naman. Okay. Sige, do you have questions before tayo mag-end? I think meron pa ito, significant figures. Let's have the last one. Sige daw, last na ito and then I'll have your laboratory after. Kindly, paisa na din, 1 to 5. Ilang atomic, ilang significant figures yan. It's 8:59 kasi maliit mo kay Ma'am MJ. Sige, let's answer this. Let's count 1, 2, 3, 4, 5, 6, 7, 8. So for number 1, the correct answer is 8. Let me recheck. Masig nalibat ko. 1, 2, 3, 4, 5, 6, 7, 8. So all are significant figures including the zeros in between. Ito, leading zeros, automatic, they are not counted. So the answer for number 2 is 1. And then ito, these are also called in between zero. So kahit may decimal pa yan, in between zero sila. So counted niya lahat. 1, 2, 3, 4, 5, 6, 7. So this is 7. And then, lahat ng leading zeros kahit pa may decimal are not counted. So we will start here 1, 2, included ang in between zeros, 3, and we also count this 0 here, a trailing 0, because remember we have a decimal point here. So for number 4, it's also 4. And then this one, ito hindi counted na trailing 0 kasi walang decimal. So the correct answer here is 5. So very good. Ayan, so we are done with chapter 1. Actually, may answer yan siya dyan sa next page. But anyway, na-answeran naman natin. Wait lang.